Hello students, welcome to my channel. In last lecture we started the light microscopy. We know that there are four types of light microscopy and already we started the bright field microscopy. So we know that for bright field microscopy compound microscope is used. Then what is a compound microscope? A microscope with a high resolution and uses two sets of lenses providing a two-dimensional image of the object is known as कंपाउंड माइक्रोस्कोप. कि कंपाउंड माइक्रोस्कोप में दो सेट्स होते लेंसेज के. फर्स्ट ऑब्जेक्टिव लेंस, सेकेंड आईपीस लेंस. ऑब्जेक्टिव लेंस फॉर्म्स द प्राइमरी इमेज ऑफ द ऑब्जेक्ट एंड आईपीस लेंस फॉर्म द फाइनल इमेज ऑफ द ऑब्जेक्ट. और ये जो दो लेंसेज हैं ऑब्जेक्ट की टू डायमेंशनल इमेज फॉर्म करते हैं, प्रोवाइड करते हैं. Let us see the image of the compound microscope. Now we are going to discuss the parts of the compound microscope. First we will discuss the non-optical parts of the compound microscope. First is the base. Base is the metallic structure and it supports the entire microscope. Then next is the inclination joint. The base and the arm of the microscope, they are joined through this inclination joint. And this inclination joint is also used when the observation of the image has to be done in sitting position, because microscope can be tilted using this inclination joint. Then is the arm of the microscope. So this arm of the microscope is also known as the limb of the microscope and it is used to hold the microscope. Then here is the revolving nose piece. So this revolving nose piece, it holds the objectives of the microscope. Next is the body tube. So the main function or main purpose of the body tube is to hold the ocular lens and objective lens at the two ends. So here is the ocular lens and here is the objective lens, which are placed at the two ends of the body tube. Then is the coarse adjustment knob. So this coarse adjustment knob is used to find and focus the image of the object. So when we want to observe the image of the object, at that time first the coarse adjustment knob is rotated and the primary image, basic image of the object is observed, and after that this fine adjustment knob is used to observe the finer details of the specimen. So this is all about the non-optical parts of the microscope. So now we are going to discuss the optical parts of the microscope. First is the mirror. So this mirror is present on the base of the microscope and this mirror consists of two sides, plane side and concave side. The main function of the mirror is to reflect the light source from the condenser towards the specimen, then towards the objective and finally the eyepiece of the microscope. Then comes the condenser. So condenser is present below the stage. This condenser consists of 2 to 3 lenses and the main function of condenser is to focus the light towards the specimen. Then is the objective. Objectives are nothing but the optical lenses of different magnifying power. There are three types of optical lenses on compound microscope: 10x, 40x and 100x. So these are the three objectives having different magnifying power. First is the 10x objective, 40x objective and 100x objective. 10x objective and 40x objectives are called as a dry objectives and 100x objective is called as a oil immersion objective. Then last one is the eyepiece. So this eyepiece, it is present at the end of the microscope or at the head of the microscope and it is used to observe the object and the final image of the specimen. So eyepiece lens is also called as a ocular lens and these also have different magnifying power like that of the objective lens, such as ocular lens of 5x magnification power, then 10x, 15x and 20x. So generally in compound microscope, ocular lens having 10x and 20x magnifying power are used. So we know that compound microscope have two lens system. First is the objective lens system which is present nearest to the specimen, and these objective lenses, they have different magnifying power, 10x, 40x and 100x. Second lens system is called as a eyepiece lens, which is also called as a ocular lens, and it is present at the upper end of the microscope, which is called as the head of the microscope. And these eyepiece lenses also have different magnifying power, 5x, 10x, 15x and 20x. Now question arises, how the total magnification of microscope is calculated. Thus, the total magnification of microscope is calculated by the formula magnification obtained by the objective lens multiplied by magnification obtained by the ocular lens. By putting the value of magnifying power of objective lens multiplied by the magnifying power of eyepiece lens, we get the total magnification of the compound microscope. Here when we take 10x magnifying power for objective lens multiplied with 10x magnifying power of eyepiece lens, we get the total magnification 100x. Like that way, when 40x is multiplied with the 10x, we get the 400x total magnification. When 100x is multiplied with the 10x, we get the total 1000x total magnification. Like this way, the total magnification of the compound microscope is determined. अभी यहाँ पे 100x का मीनिंग क्या है कि जब हम माइक्रोस्कोप में इमेज देखते हैं तो उस टाइम हमको इमेज जो है 100 टाइम्स मोर लार्ज दिखती है. दैट मीन्स मोर मैग्नीफाइड इमेज इज ऑब्जर्व अंडर द माइक्रोस्कोप. 1000x का मीनिंग क्या है कि इमेज इज ऑब्जर्व 1000 टाइम्स मोर लार्जर और magnified image under the microscope. So due to this magnifying power of the microscope, the invisible objects become visible and we can observe the magnified or larger image of the object. So I hope you understood this. If any doubt, you can ask me in comment. Thank you.